happy to have the opportunity to show our work entitled From Uncovered Genomic Elements to a Universal Cancer Vaccine. I'm Olivier Tabone, Head of Bioinformatics at ERVIMMUNE. ERVIMMUNE is a French biotech founded in 2019 by Professor Stéphane Despils, and we are developing a universal cancer vaccine. And I wanted to tell you today to show you how we moved from uncovered genomic elements to a universal vaccine using data science. First, a quick word on vaccine in cancer. The idea behind is to use the body's own immune system to identify, attack, and destroy cancer cells by stimulating a response against antigens or targets that is specific to cancer and shared across patients. So what makes a good target? A good target has to be shared across tumors and patients. A good target has to be tumor-specific, meaning it has to be present in tumor cells but absent in normal condition. It's better. Then a good target has to be highly immunogenic, meaning it has to be able to trigger an immune response. And at ERVIMMUNE, we are focusing on ERV. ERV for human endogenous retroviruses. ERV are representing 8% of our genome, and they are coming from former retroviruses infections during our evolution, and they are normally not expressed in normal conditions, but there are some evidence that some ERV can reactivate during cancers. And as coming from retrovirus, it should be recognized as a virus-like particle by your immune system as a threat. And so it should trigger an immune response. And I will show you how we developed a vaccine made of 21 ERV-derived targets. Initially, we identified 2 targets through bioinformatics pipeline that we selected for a full proof of concept, and we showed, it was very important for us, we show through wet lab experiments, extensive wet lab experiments, that those 2 targets were able to induce a T-cell response that selectively kill tumor cells. So it was very important because it showed first that ERV are able to produce an immune response, and secondly, that our process of identification of such targets is working. But those 2 targets are only covering one HLA. HLA is kind of the recognition system of our immune system, and it exists hundreds of different HLA in human population. Each individual has his own combination of different HLA, and each HLA will tend to bind to a different target. So this is important if we want to develop a universal vaccine to extend those 2 targets to more targets so that we can have a high coverage in human population. And with our vaccine, we are able to cover more than 80% of the worldwide population. The remaining 19 targets has been found through data science. The first 2 targets has been validated through wet lab, and it took years, extensive efforts, a lot of resources. Those 19 extended targets took 6 months to be identified, and I want to show you how we did that. So how do we move from 40,000 ERV fragments from our genome to a vaccine containing 21 ERV-derived targets? We used transcriptomics, proteomics, immunogenicity prediction. Transcriptomics first was used to identify the ERV that are cancer-specific in RNA. Cancer-specific meaning ERV that are highly expressed in the cancer of interest and not expressed in healthy tissues. But ERV that are identified in RNA are not, it doesn't mean that they are able to translate into protein. And these are the proteins that are recognized and bound to the HLA system. So we need to identify whether those ERV are present in proteomics databases. So the idea was to predict the part of the ERV sequence that are able to produce a protein, in theory, then to cut those sequences into short 9 amino acid sequences, and then looked for evidences into cancer patient databases in proteomics. This way, we were able to identify from 600 putative targets from our best ERV fragment identified before, dozen proof of translation. This was very important because it showed that ERV are able to translate into protein. This was not obvious before this analysis. Then final step is to predict whether those targets will trigger an immune response. And one step on for triggering a response is the binding between the target and the HLA. There are some models that exist that works pretty well, that compute a score for each HLA and each target of binding affinity between target and HLA. This way, we reduced our list to a short list of 80 targets that are with high affinity for at least one HLA type. And then we further reduced our list to 21 based on their transcriptomic profile, proteomics evidences, and prediction of binding score. So to conclude, at ERVIMMUNE, we developed a universal vaccine. Universal because we are able to cover the large majority of the human population, and we are focusing actually currently on TNBC, triple-negative breast cancer, and ovarian cancer. These are two cancers with high medical needs, unmet, that are hard to treat, and this is very important to have drugs that will avoid relapse after first line of treatment. We used, I've tried to show you how we used data science explorations to identify those targets. We had recently a green light for all the regulatory agencies. We are very close to enter into a clinical trial for the triple-negative breast cancer vaccine, and we are currently raising funds to move on the next step. Just very quickly, the perspective, our strategy for identification of target is applicable to other cancers. So this is ongoing work with our team. I spoke to you about immunogenicity prediction, but actually we are predicting the binding between HLA and target. There are a missing step that needs further model and further work. This is the step where the HLA system presents the target to the effector cells, the T cells, so that the T cells can kill the tumor cells. And this part is quite tricky to predict due to the high diversity of TCR sequence in human population and due to a lack of good quality data. And so with the emergence of new technologies such as immunopeptidomics, soon we will be able to develop a better model that will allow us to even further reduce and select the very best candidates for future vaccine. Thank you for your attention. And I'm happy to take questions if you have.
A quick question. Normally, I would say usually you build a vaccine just to avoid develop the disease, I would say, or prepare your immunology. Here, you will test it, I would say, once the cancer is already declared, and you will see if you are able to fight.
Exactly. Yeah, that's a very good point. We call it vaccine because it's related to immune response, and the idea is to train our immune system to recognize the good cells to kill. But indeed, this treatment is aimed to patients already in a very advanced stage, some with metastasis, and we will test this treatment after first lines of treatment that would not work.
And that means that you will work with, I would say, an institute of cancer to make this clinical study?
Yeah, exactly. We are actually a spin-off of Saint-Lambert, which is a hospital specialized in cancer in Lyon. Yeah.
Thank you. Great talk. It's super smart. I mean, I love the approach. As a biologist, I was wondering, how will you accept this vaccine if everything works? Because in terms of specificity, you can reactivate ERV in different conditions: oxidative stress, HIV infection, bacterial infection, ionizing radiation, and so on. So, two questions. How will you make sure that the regulatory bodies will swallow the pill, and it's only in this condition, and it will work this way, and so on? And the other question is more business-oriented. Must be very expensive to bring a therapeutic vaccine to the market. How much money are you trying to raise?
Thank you for your question. That's an excellent point. And actually, the very important part from our target ID strategy is to identify short ERV sequence that are really unique to the ERV that is shown to be expressed in our cancer. So, because ERV are repeated elements, so they have been able to duplicate in our genome, and so they are very similar. And so this is the tricky part, to identify the good sequence that is that we are sure that we are targeting the ERV that is reactivating in our cancer. So this is really part of our pipeline, this work, and this is the most important part, so to be sure that we are targeting the good sequences. And for the second part, we are actually, I'm not a good, I don't have the good terms, but we are actually raising funds, and we are expecting 10.5 million to move on the phase 1 clinical trial.
Well, good luck with that. I would love to see that on the market.
Thanks, but we are well advanced, and it's moving good in a good way.
We have also the regulatory agencies bodies. We met them, of course. Yeah, we met regulatory agencies, FDA and EMA, to introduce the product, because as you mentioned, it's ERV, so it's only expressed in human. So we need to make sure they understand the fact that we have done everything to show it's only expressed on tumor cells. And we had a very strong recommendation to move on to clinic. So we had very positive discussion with both of them, very positive.
The market is American.
The FDA and EMA, both market. Same, unfortunately, the worst cancer for women. Two of them, yeah.
And FDA, so it means you will have to launch studies in US, so it's even more millions to
So we met both of them because we start even in France, because as mentioned by Olivier, we are a spin-off, so we will use French center, and we can move on for phase 2 or even phase 3. You know, they have some agreement in between to go to FDA for phase 3. And so we will open the door by going very early to them. It's very important. They have these regulatory agencies. They really support companies like us. Okay. Yeah, exactly.